Thank you for joining us for the fourth annual Black and Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Thank you for sticking around for our second to last session of what has been a fantastic week. My name is Ade Pedro Shodi. I'm a PhD student at Cornell University, and I've been organizing with Black and Immuno for the past 3 years. For our closing keynote, we are honored to have Dr. Glenn Simmons Jr. enlighten us on his career journey and the importance of being engaged in community. I first met Dr. Simmons when he gave a seminar talk at Indiana University School of Medicine. I was most impressed by the rigor involved in his work, yet clearly communicated the goals of his research for both faculty and trainees. I don't want to give away any spoilers, but I will briefly highlight Dr. Simmons' accomplishments. He started his journey at the University of Southern Florida for his bachelor's degree, followed by a PhD at Meharry Medical College, and two postdocs, one at Louisiana State University in Shreveport, and the other at University of Texas Southwestern in Dallas. His first faculty position was at the University of Minnesota, and now he's at Cornell University as an assistant professor in the Department of Biomedical Sciences. His lab studies how lipid metabolism regulates the immune system's response to cancer. The accolades Dr. Simmons has collected throughout his journey would have us here beyond the hour, but I do want y'all to know that in addition to his scholarly work, he has been awarded the 2020 Outstanding Junior Mentor of the Year from the Clinical and Translational Science Institute at the University of Minnesota. And he has numerous awards as a spoken word artist. So let's tune in to Black and Immuno's closing keynote from Dr. Glenn Simmons. And please stick around for some networking at our final session for the week. Thank you, Dr. Simmons.
Thank you so much for that introduction. I try not to blush on camera too much, but I think you got me there. So I'm going to go ahead and share my screen here. Thank you all for attending, and hopefully this will be an enlightening discussion. So as you've heard, my name is Glenn Simmons Jr. I am relatively new to the Cornell community. I am just starting my third year here at Cornell. And today I want to kind of share with you just a bit of my journey, a bit of science, and then, maybe a little bit of career advice for all of you, no matter what trajectory you have going forward into the future. All right, there we go. So this is kind of an outline of what we're going to discuss today. So yes, I'm going to spend a good amount of time talking about myself, but I think it's important to kind of let you all know who is giving you this advice before you are able to accept it. I think that's just, common courtesy. So first and foremost, I am a Floridian by naturalization. I spent most of my formative years in Jacksonville, Florida, also known as Duval. And the picture over there off to the side is actually one of the apartment complexes that I grew up in. Some people call them projects. But I just want to let it be known that, where you come from does not necessarily dictate where you go. So for those who may still hold on to that old adage, I want to be one to very clearly dismay that. As you heard, I spent my undergraduate years at the University of South Florida, and I did my PhD at an HBCU, the awesome Meharry Medical College. And the reason I put this on the slide is that I did a lot of different things in order to kind of keep myself well-rounded, busy, some would say distracted. But again, this is all part of who I became as a young person and helped form my scientific journey. Now, one of the big things I want to kind of reiterate as we move into this discussion, I want to think of this as a discussion more than a seminar or even a keynote, is that I think it's more important to have a plan as opposed to having a map. So if you've ever had your phone do an update in the middle of, getting ready for a Zoom meeting, or if you go on to, a Google map, or back in the day, MapQuest, sometimes those maps aren't up to date when the streets change and there's a road blocked off. But having a plan, knowing where you're trying to get to, will always allow you to be agile in the moment. And so that's why I say it's always important to have a plan versus having a map. And I think as we've trained as scientists, what we tend to have is this preconceived notion of exactly how things will go. But that's not always the safest way of thinking about it. But it's also usually a way of generating a certain level of frustration as you move upon your journey. And so with that, I want to highlight that little spot at the bottom there, remembering the 6 Ps. And that's proper preparation prevents poor performance. Because if you prepare, have a plan, whatever happens, you'll be able to adapt and continue to move forward to your own destination. Now, I have a few lessons, just 3 lessons that I want to just mention that work for me. They may or may not work for you, but if you don't have, these types of lessons kind of in your back pocket, I would advise you to maybe just take these, adapt them to your own liking. So the first one is make sure that you're really careful about how you frame, your passion. Don't go into things thinking that it's always just going to be the most rose-colored version of what you've envisioned, but think about what your cause is and think about whether or not you're aligned with that cause based on the things you're doing. And so your challenge and your cause are the 2 things that I think are really important to frame early on. So know what your objective is, and then make sure that the work that you're doing stays in alignment with you as an individual so that those things don't find themselves at conflict later on and surprise you and kind of throw you off your destination, off your journey or path. The second lesson is to never forget, never underestimate the power of those people that are with you on this journey. Because no matter how successful you are, if you don't have people around you to kind of keep you honest, to keep you sane, to keep you held together, both from a mental, emotional, and a spiritual standpoint, it's no point in making all this hard work, doing all this work. And then also, as I mentioned earlier, have other things to do outside of this so-called work, outside of the science. So take up baking, take up sports. Me, I took up sports and I took up poetry, and I'm not a professional athlete, so clearly that the sports didn't really hold on for too long. But again, it helped me kind of clear my mind long enough to kind of focus and re-energize myself to step back into the laboratory. And the third lesson, which might be the most important lesson, actually comes from a Native American parable that talks about the idea of putting your energy in the place where you want to kind of build the most positive feedback, so to speak. And so the idea is that if you have 2 competing interests, if you have a negative interest or something that's negative on your mind, and you have something that's positive, and that's also on your mind, when you're constantly thinking and feeding that negative energy, well, what's going to happen? That tends to be the thing that manifests. That tends to be the thing that you're always thinking about. And so sometimes it's really as simple as just focusing on the things that you want and putting the energy towards those things. And I've seen that time and time again. So if you are trainees now, if you're graduate students, even if you're junior faculty, understanding that negative focus sometimes can be your detriment and be the one thing that keeps you from being successful. And so making sure that you're feeding the right wolf, the wolf that's going to be the one that's, hey, I'm going to go out here. I'm going to kill it. I'm going to make amazing findings in my science. I'm going to make good friends, healthy relationships, versus talking about all the negative aspects of what it means to go into these fields of science, industry, understanding that the odds are against you and that it's a very difficult thing. Like, yes, that's true, but that's true for a lot of things out there. Anything that's worth doing, there's going to be some negative aspects to it, but you have to understand that the positive things, you need to be chasing things as opposed to running away from things. And that's where this lesson comes from. Now, the reason why that lesson really resonates with me, because I, like many of you, on the path towards becoming a professional scientist, a full-time scientist, so to speak, it was always told to me that it's so hard to get, that first faculty job. It's so difficult to make that transition from a postdoc to faculty. And I saw it happen. I saw so many folks struggle with that transition. People were publishing papers, getting fellowships, but not necessarily making it to that job, getting that offer, getting that startup. And what I saw was that it seemed like we had those individuals that I saw struggling. Not only were they smart, but they were also kind of getting in their own way, in the sense that we have those maps, the way that we think things are supposed to go versus doing what's necessary to get it to go the way we need it to go. And for myself, I was determined I'm going to be faculty. That was the end of the statement. That was the plan. And for me, that meant that I ended it in Minnesota. So here you have this Florida boy moving from Texas to upstate Minnesota. Not quite my element. But at the end of the day, on the side of that door, on the outside of that lab, it said Simmons Lab. And so in 2017, after doing 2 consecutive postdocs, I was now running a small laboratory. And that laboratory basically focused on the things that I was passionate about. Because when you're training, you don't really get to do what you want to do. You're kind of at the mercy of the laboratory itself. But now with my own lab, I can start asking questions that I wanted to ask, looking at things that I wanted to investigate. And so my laboratory wanted to focus on inflammation and lipid metabolism in the context of cancer and focusing on asking questions related to health disparities. And so our laboratory has what we call a big, hairy, audacious goal. We want to cure things. We want to change and eliminate health disparities from the world. And we want to do that by taking multiple disciplines, melding those experiences together, and then coming up with novel outcomes for patients that have not been necessarily put on to the forefront of these questions. Sometimes it's hard to understand when you see these persistent health disparities in society and you have these questions that you want to ask, but the funders don't seem to be that interested in it. You see time and time again, faculty, researchers of color, not necessarily getting to get the funding that they necessarily need to get the work done. And what I want to do is find ways to get those questions to the highest levels of funding that are available, whether it's private or federal, and eliminate these health disparities by making these questions more easily answered by so many different people. And so my laboratory is a very multicultural, multi-diverse, super diverse group of individuals. And we ask these questions not because we're directly interested in, because we ourselves are afflicted, but because we're concerned about humanity at the global scale, not just one aspect of it. And because of that, one of the things that we become very kind of centered on is the idea that what we do in an environmental sense, what we do to our bodies, where we live, has an impact on how individuals end up becoming more susceptible and have increased incidence of cancer. And so over the history, relatively modern history of the United States, there's been constant shifts and ebbs and flows in what's healthy versus what's not healthy. And one thing that's become really interesting is that within our dietary history over the last, say, 100 years or so, we've shifted the relationship that we've had with dietary macronutrients like carbohydrates and fat. And what we know is that at one point or another, we really got into this low fat craze. And over time, we started to understand that, well, maybe it's not quite low fat, but maybe it's, the right type of fat. Maybe it's this fat. Maybe it's these different proportions of fat. And what's happened is that as we've removed fat from the diet, over that same period of time, we've seen an increase in things like sugar, because fat is one of those things that makes food taste good. And if you eliminate the fat, well, in order to make it more palatable, you end up having more sugar to compensate. And in the similar trend, we've seen increasing incidences of cancer over time, all cancers, and individuals ending up with cancer at earlier ages than they had in the past. And so because of that, we know that cancer is a metabolic disease, but we still have not yet really investigated just how that metabolism is being influenced by the different things that we have in our diet. And so again, we know that these things are happening because of what we're doing, but the mechanisms behind that are really murky even to this day with all of the technology we have at our disposal. And so we know cancer is a metabolic disease essentially, and because cancer is known to have a high level of glucose metabolism or highly glycolytic, we understand that that sugar that's coming in is being kind of transported, broken down, and utilized to generate energy and other macromolecules that are necessary for these rapidly growing cells. But one thing that's been understated is that sugar that's coming in, that carbon that's coming into these cells, is being shuttled off into other pathways to make other critical macromolecules, such as lipids. And so my laboratory early on became really interested in actually identifying lipids as another type of hallmark of cancer. And so shown here is the idea that when lipids are being produced, they're usually produced in the form of saturated fatty acid when it's made de novo. And so that saturated fatty acid is then converted to unsaturated fat through the activity of enzymes known as desaturases. Here we're showing a monounsaturated desaturase known as SCD1. And those saturated lipids are utilized in different aspects of the cell biology. So for membrane structure, lipid signaling, and even for energy use as well. And why that's interesting is because when you're starting to understand that the lipid component is another place where all of that extra energy is going. It becomes an obvious thing for those who are interested in the cell biology of cancer that we need to understand why these lipids are doing what they're doing, especially when you have cancers that are known to be fatty in presentation. And so our lab really became interested in just asking a very basic question. Does the level of fat or unsaturated fat in patients affect their outcomes? And can we take that knowledge and utilize that to improve therapeutic outcomes in these individual patients? And so to start answering this question, we accessed the Cancer Genome Atlas lung adenocarcinoma data set. And within this data set, there's some 400, 500 some odd patients. And we saw that the expression of that desaturase gene, SCD1, is significantly lower in patients that have lung cancer. But if you look within those patients, the patients that are at the highest level of expression for SCD1 versus the patients at the lowest level of SCD1 in the tumors actually have a very different outcome in terms of their overall survival, which indicates that even though the overall expression of SCD1 is lower in the cancer, there may be some kind of adaptation once that cancer manifests that then creates another kind of stratification within this tumor, within these patient populations, which could ultimately affect how we're going to implement therapeutic approaches later on. But again, this is something that has yet to really be utilized in terms of the standard of care. And so within our laboratory, we took a standard lung cancer cell line and we performed a process known as delipidation. So we essentially lipid starved the cells briefly overnight and then reintroduced different types of lipid, either monounsaturated, polyunsaturated, or saturated fats. And it was in the presence of both the unsaturated fat species that we saw that a specific subset of proteins related to the immune response were being upregulated significantly. And if you can look over here in this volcano plot, you can see that one of the proteins we kind of became pretty enamored with was a protein known as HMGB1 or high mobility group box 1 protein, which is a protein involved in inflammation. Now, HMGB1 is actually a pretty interesting protein because it's normally secreted from cancer cells and leads to the recruitment of immune cells, but it also has a role in suppressing the immune response. So it has kind of this duality. While it's being secreted, it recruits certain immune cells, but it also leads to the inactivation of T-cell mediated immune responses. And so what we saw in the data shown here, kind of through multiple experiments, that the presence of unsaturated fatty acid stops the protein from being secreted in a very significant fashion. You can kind of see that here. You see protein retention in the immunoblot shown here. And then in the ELISA data here, using two different cell lines, you can see that the amount of HMGB1 protein released is significantly suppressed, both in the A549 and the HCC827. We can actually show that same effect is present even when you express an exogenous protein, which indicates that this isn't an effect just based on gene expression or something like that. We've done the PCR. PCR shows that there's very little effect on the mRNA expression of that particular gene. So this is all at the protein shuttling stage. And if you take those same cells and you look at the activity of NF-kappa B, which is a major transcription factor involved in inflammation, you can see that the NF-kappa B nuclear binding is significantly decreased in these cells. And that comes to find out it's actually a paracrine type of response because the expression of the PDL1 gene, the mRNA for programmed death receptor ligand 1, which is a major immune checkpoint target, is actually driven by NF-kappa B. And so we can see that TNF alpha, which is another NF-kappa B responsive gene, is suppressed by the presence of unsaturated fatty acid here in the form of oleate, both in our direct treatment or in our delipidated treatment. And if you do a dose-dependent dose curve with oleic acid, we can see that PDL1 levels are decreased as HMGB1 levels are increased in the A549 cells. Now, what makes this really interesting to us is that this effect is not just intrinsic on the cancer cells, but there's actually a paracrine type effect when you co-culture the THP1 monocytes, or when they're differentiated into macrophages. You can see that the conditioned media from the A549 cells actually leads to a decrease in PDL1 also on the monocytes as well. which indicates to us that this is an effect that's happening within that tumor microenvironment. And so that led us to kind of generate a hypothesis that shows that the expression of HMGB1 from cancer cells is actually inhibited by the presence or the levels of unsaturated fatty acid. Now, normally in the absence of unsaturated fatty acid, or when those levels are sufficiently low, HMGB1 has the ability to signal both to monocytic cells, but also to stromal fibroblasts within that tumor. And that leads to essentially a immune suppressive phenotype, which restricts the ability of cytotoxic T cells to then target these cancer cells, allowing those tumors to grow larger and become essentially immune to checkpoint blockade therapies, which is really important because that's currently one of our major breakthroughs in the treatment of cancer. In order for us to take advantage of this in vitro, we've developed a 3D bioprinting model in order to investigate this. And so we, in our lab, have a bioprinter where we take both the patient tumors as well as patient-derived cancer-associated fibroblasts. We disaggregate the tumor in order to make a single-cell suspension, create what we call a bioink that actually consists of a methacrylated gelatin, so that allows us to cross-link the gelatin later with the cells embedded. And we create these kind of what we call tumoroids or assembloids that will have all the different cell populations in a three-dimensional space. And so here you can see just a cell-latent droplet of just A549 cells. And here you have an A549 spheroid that's embedded within the hydrogel. And what you can't see here is that there's actually fibroblasts all throughout the media outside of the spheroid. And you can see right here, you have a necrotic core within that spheroid. Now, this gives us the ability to start asking more questions about what the composition of the nutrient space around these tumors, what the effect that nutrient space has on the efficacy of immune checkpoint therapies or even chemotherapy, without having to rely exclusively on getting things inside of PDX models, transgenic mice, or even getting samples directly from patients. Because by taking a patient tumor and embedding them in these tumoroids, we actually can multiply a patient's sample several fold and allows us to test multiple modalities simultaneously in half of the time that it would take us to do this if we were doing this in an animal model. Now, why is this all important? Because again, immunotherapy is such a revolutionary treatment to help us cure and eliminate cancer. Well, unfortunately, particularly with lung cancer, lung adenocarcinoma, almost 80% of the patients will actually either fail or eventually fail to respond to immunotherapy. So that means that even though this therapy exists, most of the patients are not going to benefit from it. And that means that there's something that we're still missing. Even though we do everything based on these protocols, based on the clinical trial data, there's still a major gap in our understanding. And so what really got us excited in our laboratory was that we know that there's this thing called the obesity paradox. This is a situation in which patients who actually have elevated BMI are doing much better than patients who have normal or low BMI. Now, for those who are kind of in the more translational space, or even those who are in the medical space exclusively, we know that obesity tends to be associated with poor outcomes across the board. And for those who do cancer work, the idea of obesity as being a pre-existing condition that tends to lead to cancer is a well-known fact. But the obesity paradox actually kind of turns that whole idea on its head, because here we are looking at patients that are receiving chemotherapy, immunotherapy, and we're watching those patients that have the elevated BMI perform better with treatment. And we think that that has something to do with the level of adiposity in the individuals, but we're not seeing that data because that's not the type of data that we collect when we're doing normal clinical visits. And we want to change that. So to wrap up this bit of science here, I just want to kind of highlight that we have some really interesting observations that show that fat, unsaturated fat in particular, regulates the expression of these key targets of immunotherapy. And that we really have to revisit this idea that inflammation, immune suppression, and the amount of fat in patients is always going to be kind of a one and done situation, that we know all there is to know. It appears that there's a lot more nuance there, and we need to dive much more deeply into the information, start doing more personalized medicine approaches with individual patients so we can understand what's happening and come up with strategies for those patients to survive and not become the 80% that don't seem to respond to therapy. And also, Again, we're super excited about the idea of generating this platform for utilizing patient-derived samples in 3D bioprinting modalities that will allow us to test multiple therapies on relatively small amounts of sample. Because again, when you're dealing with getting a hold of patient samples, especially as a basic scientist or someone who works exclusively in a lab, that can be a major roadblock to really getting innovative information that could then ultimately help improve the outcome in patients. So we're looking to develop, clinical interventions, but we are working right now to develop this platform so that we can generate the data that's necessary to make that pilot study significantly more feasible in the future. Now, one of the things that I want to say before we pivot is that this bioprinting platform is in many ways a necessity that was born out of a very bad situation. So most of us, everyone should have had to adjust their lives somewhat based on the COVID-19 pandemic. And my lab and the individuals that worked in my lab back at the University of Minnesota were no exception. And when the pandemic really started to set hold, our lab effectively shut down. And during the process of shutting down, what happened was that all of our cancer patients that were enrolled in our studies stopped showing up for treatment because they weren't necessarily going to have enough tissue to send to us to our laboratory. And our lab, as we were kind of waiting for them to tell us time to lock it up, time to shut down, we're like just praying, like, please, we just need one more sample. We just need a little more data so we can get this grant out, just a little more data. And that's when we came up with the idea, actually, of using bioprinting as a way of taking a limited sample and, basically managing to get as much out of those samples as possible for as long as possible. Because again, we're not in the clinic, we're not in the hospital, so we're at the mercy of whether or not the clinicians and the pathologists think there's enough tissue. And over time, as the pandemic went on, those surgeries became less and less to zero. And when we hit zero, we were done. Luckily, with the bioprinter, we had new ways of, utilizing the samples we already had. And the other thing is that the pandemic also highlighted something that many of us that are highly melanated, we experience, the idea that health disparities in the United States and abroad were made worse thanks to the pandemic. So the issues of access, the issues of availability of resources became that much more obvious to many of us thanks to the pandemic. And health disparities isn't something new within the United States. It's actually been studied for over 100 years, and we know a lot of the factors that contribute to it. Your living conditions, your income, all of those things play a major role in how health disparities manifest. And thanks to COVID-19, now the numbers that are here are based on when we initiated our study, but we knew that there are so many communities of color, whether immigrant populations, native communities, African American communities, all were finding so much of that that worry and that angst just being amplified hundredfold thanks to the pandemic, because no one knew anything and there was just not enough resource to go around that people were comfortable with giving to folks that weren't already in the know. And so my laboratory and the University of Minnesota Medical School in Duluth, we were trying to figure out, well, what could we possibly do? So I was at a small regional campus of the University of Minnesota, which basically means, as opposed to being in Minneapolis, I was actually up by Lake Superior. So we were what we would call rural adjacent. And we knew that we didn't have the same kinds of resources that they had in the Minneapolis area. We didn't have a major, super big hospital with a big research infrastructure. We were just little medical school with a bunch of researchers that wanted to do something. So my laboratory thought, well, what is it that happens with COVID that we could take advantage of? What are the qualities of it? And luckily, SARS-CoV-2 was similar enough to SARS-CoV-1 that when we started asking these questions, we realized that, hey, well, the virus, or at least the viral particles or aspects of the virus, seem to be present in individuals for a significant period of time, but they actually start showing up pretty early as well, depending on how you're looking for them. And that became like a really interesting conversation around our table as our laboratory and some of the other folks in my department were trying to figure out, well, what can we do? And so I proposed that, well, what if we did things similar to what they had already done with SARS-CoV-1? Let's start looking at the excrement. Let's see what's coming out of people. And of course, that wasn't the only time it had been done, because for decades it had been done with other viruses as a way of keeping track of how the virus has been eliminated for things like polio. So it was part of a surveillance mechanism for polio vaccines for many, many years. And in 2020, in the Netherlands, a group had actually started looking at the municipal wastewater in a couple of cities in the Netherlands. And then there was a group in the Boston area that was also doing something really similar. Now, what was interesting about both of these groups is that they were kind of doing this kind of high level thing. They were in these major metropolis areas asking, can we see it? Well, what I was concerned about was, how can we leverage this for places where they can't get testing done? Can this be utilized to kind of supplement or even completely outsource the idea of requiring individuals to find their way to a clinic that they would end up getting turned away from? Because there were countless stories like that, even in the community that I was living in at the time in Minnesota, where their testing facility was somewhere up on a hill, but a lot of the people who lived in any kind of subsidized, housing, they were down at the bottom of that hill in the wintertime. So trying to get them up that hill required that they had their own vehicle, which they may not have, get on public transportation, which at minus 20 in the wintertime, you're probably not going to do that. And so you basically force people to either do things that weren't necessarily safe, which would be like carpooling and things like that, to go up here and get tested, so they wouldn't even do it. So we wanted to figure out a way of getting this technology, getting these types of questions answered without limiting or cutting off people from these resources. And so we developed this hypothesis, that SARS-CoV-2 could be measured in the wastewater, and then maybe we can estimate the number of people that are even infected in a community without doing that individual testing. And so to do that, we also wanted to make sure that our method of doing this was completely different than what other folks were already proposing. And so some of the laboratories early on in the pandemic came up with methods that required you to have, a couple of gallons of wastewater. Now, again, let's just be real, wastewater, you don't want a whole bunch of it. So we were trying to minimize how much of this we had around, but then also we wanted to make sure that we developed a technique that could be utilized in any site that had basic access to laboratory equipment. Do you have a centrifuge? Do you have a basic PCR machine somewhere in your city? Now, like a lot of states, you have state universities, and in a lot of the state universities, they would have, a real-time PCR. They may only have one. It may be out of date, but they would have it. And so we wanted to develop the method and then allow that method to go out into the communities across the entire state of Minnesota and maybe across the whole country, just giving it out for free. And so as you can see here, there was nothing super fancy about what we did, but the idea was that everything would be at a scale that could be managed by a really small team of folks that had basic biological understanding. And so when we set out on this, we made one phone call, or I made one phone call, and I called the wastewater treatment facility in Duluth, Minnesota. That phone call led to a phone call to 10 other sites. That phone call from the 10 other sites led to a phone call with 20 other sites. And by the time the study was completed, we had 44 cities across the state of Minnesota that were all encompassed within our study. And as you can see here, if you look at panel A, this is the amount of the population for each region that's actually served by a wastewater treatment facility. And then if you look over here, this is the amount of coverage we actually had based on the facilities that agreed to work with us. So we had a good amount of the population of Minnesota that were represented in our study, and we could kind of batch them based on where they were located. Now, when we started collecting data, we were kind of on our own. No one was really willing to accept that what we were doing was going to work. That was the first thing, which makes someone like me very nervous, because as a junior faculty, I had to step out, I dare say on faith, to get this to work. But during the whole period of, my laboratory, my technicians trying to get this to actually start working, we were seeing the frequencies of these clinically diagnosed cases throughout the state over this entire time. We didn't start collecting samples until around here. So I would say late May. And as we started collecting samples, we became really interested in determining whether or not what we were seeing in our samples in the lab, not only were we worried about them being positive, but we were worried about whether or not our data looked similar to this data shown here based on what was showing up in the clinics across the state of Minnesota. What was also really interesting to us was whether or not we could see trends that followed what was happening based on what recommendations were coming from the governor's office. So you can see here we have indications on when the stay-at-home orders were lifted, when they started letting people go back into restaurants, when they instituted the mandatory masking. And you can see it almost looks like some of these things have kind of a preceding relationship to when the spikes start to happen. And you can see, like, even here, when the mandatory mask order, even though things started going up as people started going out more, the masks show up and things start to trend down again. So we thought that, okay, this is pretty interesting because this is coming from our data. We can kind of see that as things are happening at the political level, we're seeing what's happening at the biological level. But what was really important for us was to figure out how to translate all of these, read counts or CT counts based on our PCR analysis, making this translate into people, making this something useful so that the clinicians and the public health folks could actually do something with the information. And so what a lot of studies up until this point were doing, they were basically just saying, hey, we got some numbers, look at this. Ooh, it goes up, it goes down. But as you can see, if we look at central Minnesota comparing to metropolitan Minnesota, so this is going to be like the Twin Cities, Minneapolis area. For those who've been to Minnesota, that's the most densely populated area in the state. So as you can see, it looks like, wow, there's so many new cases. This is where you need to focus all of your energy. So throw all the money there, try to stop the spread in those areas. And compared to the new cases in central Minnesota, some stuff happening. But when you start to normalize that data, you see that central Minnesota was getting the mess kicked out of it. Comparatively, that spike is way more significant in central Minnesota than it was in the metropolitan area per 10,000 people, which indicates that without normalizing the data, you may get a completely different conclusion than what's actually happening. And so when we started to collect our data, we decided to come up with what we call an index to take all of the data and then normalize it based on the amount of essentially a highly abundant RNA virus that's in the human diet. So this is something that's used in the environmental science area a lot, where they use mild mottle virus, which is a tobomo virus that is common in pepper plants, and it does nothing to us, but it shows up in our excrement, and it allows us to keep track of the quality of the water over time. But for our purposes, it allowed us to normalize all of our data, not just based on people, but based on the amount of this virus that was present in the water. So we were able to take all of this data, which kind of had a lot of noise in it, and we were able to clean it up and bend the data into this quantile setup that allowed us to make more sense of what we were seeing over time. And so as I start to close out on this section, you can see that over time in the gray area, you can see the total number of new cases. This is coming from the dashboards from the health department and the clinical cases. And you can see what we were seeing based on the N1 and N2 probes that we were doing our PCR on. And you can see that it almost looks like our curves are showing up before the curves in the clinical cases in the hospital. Now, at first, that doesn't seem all that interesting until you do what we call a lag analysis on that same data. So you can do N1 and N2 separately. And if you do this lag analysis, you actually see that our data, if you do the lag analysis, actually precedes the data that's showing up in the clinics by 2 weeks or more, which indicates that our data is predicting what's going to happen in the clinic, which even though we were setting out to really come out with a measure of how many people were infected in a given area, the mathematics on that was a little bit beyond my team. But what we ended up walking away with is a predictive tool, because the Melvin index is actually coded. And every time we get our data in, we throw it into this code, it spits out the projection. And that projection ended up getting us a few conversations with the governor and a little bit of notoriety within the university. And again, I failed to mention, but at this time, I was not a tenure track professor. there wasn't a certain amount of just given prestige to the fact that, hey, I want to go and do this thing. This was something that me and my collaborator at the time just said, hey, we want to stay in the mix. I don't want them thinking that we don't know how to do anything just because we're at a small campus here in Minnesota. And we ended up coming out with this really cool model. So to summarize that particular project, so we ended up becoming essentially a part of the state action plan. So the Department of Health sponsors that work. So it has sustained funding going forward for I don't know how long, because as you all know, at this point, I'm no longer at the University of Minnesota. I turned over the project into good hands. And again, I mentioned we expanded from what we started out with was just 19 cities. And by the time I left, we were up to 44 cities across the state. What was really cool is that because we were essentially operating from a grassroots standpoint, the relationships that I had with all these different cities around the state of Minnesota, cities and towns. So our largest city was Minneapolis. So that's like a population of roughly 2 million that we were collecting data on. And then our smallest catchment area was around 500. Okay. So 500 to 2 million. And each of those wastewater treatment facilities, I had a conversation with and really got to understand where they were concerned about, well, what's going to happen if this town hears about what we got going on? Are they going to try to take business away from us? I mean, it was some really involved conversations about things that I had never been trained to talk about as a basic biologist, biological researcher. But what happened because I had a biological question and I put myself into that space. At this point, that whole project, that whole workflow, the shipping and all these different things that we had like mapped out are all connected now to a next gen sequencing workflow that allows us to now look for novel pathogens while we're still monitoring COVID. So we basically took an entire system that we developed that involved generating APIs and having, prepackaged labels and shipping containers, working with the mailroom at the college in Duluth. All of that was set up by us. So all the samples came to us and went back out. We actually processed about 800 samples per week with a team of about 4 people. And really no one was working at that institution before the project started. So in a way, by doing this work in the pandemic, I don't know how many folks experienced layoffs and things like that, but we actually had a lot of people end up not getting, brought back. And we were able to hire people in the midst of the pandemic to get this thing going. And also we were able to kind of keep these folks at these wastewater treatment facilities, keeping them even more essential than they already were to the various communities that they were in, because these people were now the touch point as to what was happening with a major, pandemic scare. They were having conversations with us, and then they were talking to their mayors. They were talking to their city council. So I think it's really important to understand just how important we are as scientists, regardless of your discipline, in kind of moving things forward and quelling some of these fears when people don't understand. We're trained to understand things that aren't understandable. We ask questions that no one's asked. And I think it's important to remember that that is our superpower. And that's something that we can always bring to the forefront. And it doesn't just have to be in the midst of crisis. That can be an everyday thing. So in conclusion, for everything, including the cancer work, I think what I would say to any young scientists that are on this in this talk right now is that you have to become comfortable with making decisions without having all the data. You make the best decision with what you have in hand. And the add-on to that is don't go back and then look at that decision now that you know something new and just like, oh man, if I'd only knew. It's like, no, you made the best decision that you could with the data you had. Okay. And then also understand it's okay not to know everything. Know what you know and accept there's some things you don't know, because that part will drive you up a wall. And I think life is too short for us to be constantly kind of looking in the rear view in that way. And then also be willing to go out there and figure out what you need to get in order to get what you want. But also remember that I'm sure many of you are probably way smarter than I am currently or I ever was. But understand, like, no matter how smart you are or how capable you are, you should never try to do everything by yourself. Because I think that ability to bring people in, that is where success lies for those who are consistently successful. It's not about going out there and being a maverick. It's about going out there and being the best team player that you can be. Sometimes you're the quarterback, sometimes you're on the offensive line, sometimes you're the running back. If I could come up with another analogy, I would, but hopefully everybody gets a football analogy. Okay. Now, with that, I have to thank my amazing team here at Cornell. I've had an amazing transition from my time at the University of Minnesota to here at Cornell. My lab manager, Nicole, is probably the number one asset that I would say that I was able to come in contact with. I have some amazing students. Ari is my graduate student whose work isn't highlighted here, but she's great and doing her A exam soon. So if I have some prayer warriors out there, I need your help because I'm nervous. But Lord knows, I think she's got it. The paper or a lot of the cancer research was done by Brianna or Bree, who was a graduate student at the University of Minnesota. She just completed her master's. I would love to thank all my collaborators, both here at the College of Veterinary Medicine, but also at Upstate Medical Center in Syracuse. I'd also like to thank NCI for funding my transition here, as well as my startup, which was instrumental in me being able to make these hires that I've done recently. Now, I would be remiss to not talk about the amazing COVID wastewater lab team. Dr. Richard Melvin, who was my co-conspirator in getting that project off the ground, who's now in Australia at a university there. I believe he's at Monash. No, he's at LeTourneau in Australia. Shannon was my lab manager. She was the first person that we hired, and she coordinated the rest of the team. Daniel, Ty, Katie, Stephen, and Adele. She made so much come together with no experience. And again, it's something about being in those moments where you have to perform that you really know who you want to have in your battlefield. And I would say more than anything, nurture relationships full time, not just when you need them, not just when you need a letter of recommendation, because when you get into those moments that are stressful, that are trying, it's good. I would say it's essential to know who you can actually rely on, who you can ask to take that load off your shoulders, and not be surprised when folks can't show up the way that you expect them to. A lot of times that's because you've never actually had to test that theory. But long and short, the funding that was able to, be responsible for this project, it was trying initially, but luckily to the funders of my initial career development award through the CTSI at the University of Minnesota, they are the reason why that project got off the ground. And eventually other people saw the light and supported that work. And I'm so thankful for them. And I'm especially thankful for the Minnesota Environmental Science and Economic Review Board, because they are the ones that are responsible for connecting us through the various wastewater treatment facilities. I think, again, that relationship piece is so important with science because science is becoming more and more interdisciplinary. I know there was a session earlier talking about AI and ML, and honestly, I couldn't have that conversation with anyone and be knowledgeable, but I know that for my work in the future, it's going to be essential to start having these conversations about how to take big data and make it make sense when I'm studying a single mechanism in a single type of cell or a single type of cancer. And I definitely would love to hear more from all of you. And if there's some elephants in the room, some things we need to discuss, I am more than willing to have that conversation. Thank you.
Thank you, Dr. Simmons. Yeah, I was doing a full talk all there by myself. Anyway, I'm looking in the chat, and it seems that a lot of people found what you had to say very inspirational. And I'll get started with the first question. That it was a great example of changing paradigms in science. What challenges do you see in changing the paradigm of fat being bad in health?
So that's a really good question, and I think I spoke to that really briefly. I wish I could have like a whole conversation on this. And this is, again, back to that point of talking with folks that are in different disciplines. I think when we start looking at personalized medicine, I think we're going to have to start understanding, well, what type of fat what proportion of fat are we talking about? Under what condition, right? So within that idea of the obesity paradox, there's this concept of metabolically healthy and metabolically unhealthy obesity. And ironically enough, it looks like the metabolically unhealthy people are the ones that are responding better. And we suspect that that's largely because the proportion of certain fats are different in that population than it is in this other population. So I think we need to start doing more personalized medicine on the patient history side of things. When you show up, if we could do some kind of metabolomics off rip, we could probably change the game really soon. But the fact of the matter is that it's cost prohibitive. So I'm sure there are people who have that kind of concierge service. They walk into their doctor's office and they're like, yeah, so breathe into this. We know what everything that you metabolizing right now. You need to stop doing this. But when you think about the folks who visit the doctor only when they're actually sick, mm-hmm. they may not ever get that because you're coming into an emergency room type of situation. You're not coming into a UT Southwestern. You're not coming into a UCSF type of research institution. You're coming into a neighborhood clinic. Mm-hmm. So I think that's the biggest challenge, is making sure that everyone gets access as soon as possible. There's always going to be a divide between who gets it, the early adopters, and everybody else. But I think as soon as we start that process, the sooner we'll get to the point where we can implement more omics in regular treatment.
Right. Thank you for that. Mm-hmm. I think at the beginning of your talk, you mentioned 6 Ps to kind of like get yourself acclimated to kind of like a life theory or being in science theory. Could you mention those 6 Ps again?
Yeah, absolutely. I don't think I can say out loud where I got that from, but but the idea is proper preparation prevents poor performance. And again, it's the idea of planning versus having a map. We're taught in education, especially in, quote unquote, the Western world, that there's a linearity to education. And because of that, you have this map. It's like, okay, when I graduate here, I'm going to go here. And when I graduate here, I'm going to go here. When I get my PhD, I'm going to get a postdoc. And it's like that linearity is actually the thing that's setting up a lot of people to fail, right? As opposed to saying, I want this outcome, however it may come, right? So for myself, I said, I want to be faculty. And I told myself, don't get married to a location. Don't get married to an institution. Mm-hmm. You want that job. Or like for students, I tell them, it's like, your job is to get the degree. It don't matter where you get it. It don't matter how you get it. If it take you 4 years, it'll take you 7 years. Do you have your letters? Yes. All right, move on. Because there's plenty of people who don't have it at all. So if you want to keep comparing yourself to someone else's plan, it's almost like when you try to edit your life by looking back. It's like, well, if I'd have did this back there, it's like, but you didn't have that information to make that decision, right?
It's so true.
Proper preparation prevents poor performance. So once you're ready, when the opportunities show up, you got it. It's like, well, I know I want a faculty job. Somebody says, hey, I'm going to give you this faculty job. You're not thinking about it. You're saying, I wanted a faculty job. Now, if you start saying, I want a faculty job and the school colors got to be, this, that, or the other, you find yourself hemming and hawing, and that opportunity goes to the person who was ready to take it.
That's true. Yeah, I think in the same vein, maybe like not putting limitations on your interest and being more open to any way that might look. You said that should trainees now be like advocating for their particular interest, or is it like, I'm in the space to learn and I just absorb everything and put out a product?
I think both. Okay. I think both. So, and that's the thing, I think we want things to be simple, and they're not. I think the only time it's simple, and I'm gonna tell all my business today. I have children. At this point in my life, nothing is simple because I have tiny humans that I'm responsible for. But when you're a student, you kind of want it to be like, I'm a student. This is all I am. But I tell people, like, you're an adult, which means that sometimes it kind of sucks, but you have to make hard decisions, and you're responsible for those decisions. So if you are training to get a product, you're training to get a product. That does not change. That becomes kind of the background. That's like the base of your cake. You need the cake. Now your icing is you doing the exact thing you want to do later. But the cake doesn't change because you can't graduate without the product.
Right.
But you may know that I want to go into a particular industry that may not even exist. Mm-hmm. So I always advocate for students, if your primary mentor, as long as they're not your antagonist, Find someone else that's the better person to help you in that space. If it's not your primary person, because if you have an antagonist, you need to deal with that, right? But if you have somebody who's generally supportive, but they just can't help you in that area, get another person, add them to your team, and keep moving. And those things have to happen at the same time, because if you wait, that the 6 P's aren't being implemented if you wait. Right. Go ahead and start planning. Like, hey, I need to go to this conference by this time because it'll give me enough time to figure out if I really want to do this thing later.
That's a solid point. I want to give those in the chat or in the audience a moment to post their questions as well. But I have another follow-up in case they're still thinking. So we've talked about health disparities, and I feel like that's almost the polite way to get away from saying the outcomes of racism. And so I'm wondering, is it something that you mentioned maybe like in verbal, but like when you're writing a proposal, you just focus on the basic science of whatever your question might be. But what is your insight on that?
I see what you're doing here. No. So in my slide, I don't know if I changed it too much, but no. So when I say health disparities, I say that health disparities is actually much broader than the idea of racism. It's much broader than that. And again, being someone who's lived in spaces and been amongst rural populations it's way beyond that. Because if anything, health disparities is probably more of a byproduct of capitalism than it is racism. Racism becomes a product of capitalism. Don't get me started. But but because I do work kind of on the social science side as well, I can say it. I can call it what it is.
Yeah.
But when I'm looking at it from the standpoint of knowing that access in a lot of different ways, like if you tell me like a European American farmer in upstate New York has the same issue as a Black single mother in Mississippi, I can't call racism on it because it's not technically that. But it's still access. It's still an access issue because they still may show up at the same hospital and get the same kind of like side eye, right? And so I think it's nuanced, and you have to be willing to have all of the conversations, because sometimes it's multiple things. Sometimes it's just one thing. Sometimes like, I just don't think you can pay. Now, why I don't think you can pay, that might be the nuance, right? But just the ability to pay. Now, if we were in a situation where things like, again, thinking omics being a part of standard of care for everyone, and it was already paid for by the government somehow, at that point, and nobody actually pays out of pocket, like that would eliminate at least the conversation like, oh, this is capitalism. It's like, oh, maybe not, because everybody's paid for already. So now you can call it what it is.
Right. Okay. So I'm not seeing any more questions in the chat. And we're awfully close to the end of our session anyway. And so I just want to thank you so much for joining us this afternoon for me and other times for others. And really just like, I hope everyone had an appreciation for what you had to say, because your story, it's right, it's not linear, but at the same time, you're still able to reach your own like personal goals and reach these successes that wouldn't happen if you just follow the path that everyone else follows. So thank you so much. And are there any good resources on ethnicity and disease epidemiology? I'm sorry, I just jumped right back into it because
No, it's fine. I saw it pop up as well. I mean, there are several, but I would always recommend, so for myself, not being a preeminent expert in that, because even when you talk about ethnicity, again, you become even more nuanced, right? Because depending on where you are, even within saying African American, we are a cornucopia of different things.
Yeah.
And so I think you would probably want to look at some of the journals that are kind of more in that space. I think Meharry actually has one that's out of there. It's like the Journal of, gosh, too many letters, so I can't remember it right now. But if you were to send me an email, I promise you I would connect you with the folks. I think the editor is actually someone I know right now, if you're really interested in that, the person that made that comment.
Yes, Medina, I can share the email with you. So yeah, I think we're gonna close it. And thank you so much for joining us, doctors. That was Dr. Glenn Simmons for our closing keynote for Black and Immuno.
And thank you so much to the ASL interpreters. I saw y'all. I appreciate you, because I know I was saying a lot all over the place. So much love.
It looks like we're going to have networking at the end of this, so stay tuned for that. Otherwise, we're finished.